Thanks so much to everyone for coming. We're going to be talking today about Parkinson's disease. I'm Michelle Fay Cortez. I'm a writer with Bloomberg and Bloomberg Businessweek. I've been writing about healthcare and technology for the past 22 years, all for Bloomberg, and it's been a very long and interesting journey. But Parkinson's disease is definitely one of the intriguing and challenging diseases that I've been writing about over these past few years. We're almost on 200 years of discovery of Parkinson's disease. In 1817 is when we first really started learning about the condition itself, and we still don't have any drugs really to slow the progression, and we're still trying to figure out about the details of the disease itself. And so I'm just going to have everyone here introduce themselves and tell you guys who they are and what their role is in Parkinson's disease research. We'll start with Frank.
Yeah, so my name is Frank Bennett, and I'm head of research at ISIS Pharmaceuticals. And we currently have a couple of programs in late-stage drug discovery that are developing therapies for Parkinson's disease.
I'm Michael Schwarzschild. I'm a neurologist at MassGeneral Hospital, physician scientist, and also lead the Parkinson's Study Group clinical trial consortium for Parkinson's disease in North America.
Clemens Scherzer. I'm a physician scientist. I lead the Neurogenomics lab and the Parkinson's personalized medicine program. I also co-founded the Harvard Biomarkers Study and program. We are working to change the way we look at Parkinson's and the way we treat Parkinson's using genomics and developing a personalized, precise medicine for it.
I'm Todd Sherer. I'm the CEO of the Michael J. Fox Foundation for Parkinson's Research. We're a nonprofit foundation focused on Parkinson's disease, and we lead an aggressive research agenda supporting about $60 to $70 million of Parkinson's research and therapeutic development annually.
So Michael, I think we're going to start with you, perhaps giving an overview of where we are with Parkinson's disease.
Sure. So just a thumbnail for a few minutes on the disease in the context of some of the other neurodegenerative diseases you may have been hearing about in the last couple of days. Parkinson's is a neurodegenerative or brain cell degenerating disease. Clinically, in its classical manifestations, it entails progressive development of slowness, rigidity of muscles, often the characteristic tremor of the hand, usually on one side, when the body's otherwise at rest, all typically converging to present a clinical diagnosis of Parkinson's disease. Parkinson's is the second most common neurodegenerative disease, second to Alzheimer's, about 5-fold lower incidence and about an order of magnitude more frequently occurring than ALS and more so than Huntington's. Since the first published description by Dr. Parkinson's that Michelle mentioned some 200 years ago, we have come a long way in terms of what we now understand about the disease, particularly in the last couple of decades. So about over the last 100 years or so, we've gained an appreciation of where in the brain the classic motor deficits that I mentioned, what changes give rise to them. It's actually in the upper part of the brainstem in a region that macroscopically looks like a black substance, and hence the name the substantia nigra, which degenerates early in the disease with classic pathological features that have become to known as Lewy bodies. And about 50 years ago, it was discovered that that black substance, being a breakdown product of the chemical signal characteristic of those degenerating brain cells, dopamine, degeneration, with it comes the loss of that chemical signal or reduced levels of dopamine in the substantia nigra and the brain region to which it projects, leading in short order to identification of dopamine as the sort of missing signal that lubricates normal movement, such that the first and arguably still the greatest advance therapeutically in Parkinson's disease was the discovery that a precursor, building block of dopamine, levodopa, and other so-called dopamine replacement strategies can markedly improve the symptoms of Parkinson's disease. To be clear, that's only partially the symptoms, and there's no evidence that it directly affects the underlying disease, such that people often have the impression that levodopa only works for a set number of years and then loses its effect, whereas in actuality, levodopa remains a very effective drug for almost all patients with Parkinson's disease, yet over years as the disease progresses, there's more dopaminergic cells lost, and as we'll be talking about, there are more non-dopaminergic cells that also degenerate, leading to other features of the disease. So that's where things were about 20 years ago, until there was really a major shift with the advent of a number of genetic clues sort of changing the landscape of how we view not only the biology of the disease, but the therapeutic targets that are emerging, and interestingly, even clinically, what the disease is like. And I'll just mention genetically, just to be clear, Parkinson's disease is not a classical or monogenetic disorder. It's not like Huntington's disease, where almost all of the disease onset is attributed to a particular gene. Yet, as we're increasingly appreciating, there's a number of genetic influences that work together with non-genetic ones, presumably environmental factors that are still very, very poorly characterized. But those genetic clues have been the substrate of a tremendous amount of advances that are leading to therapeutics that I think we'll be talking today. And I just lastly wanted to say that in an interesting way, the genetic advances have also allowed us, fostered a greater, more holistic understanding of the clinical aspects of the disease, with the appreciation that one of the lead genes in the biology of the disease, alpha-synuclein, is present not only in the pathology of the substantia nigra, in those Lewy bodies I mentioned, but as it turns out in other brain regions as well, even actually seems years before, below the substantia nigra, in brain regions before the motor features develop, and then as the years progress, higher up in the hemispheres of the brain, leading to new insights into theories about how the disease may spread through protein going from one neuron to the other, but also clinically to a greater appreciation of what patients know quite well, and but we now incorporate into our broader view of how it's many more structures, many more systems other than just the dopaminergic motor deficits of the disease that are involved. So with that sort of broader view that's developed in recent years, I think we have seen the clinical picture more broadly, as well as the biology, and that's led to a lot of, well, challenges in how we approach the disease, but also a lot of opportunities for drug development. I think we've changed a lot, and that leaves a lot to talk about, so I'll stop.
Right, thank you for that. Certainly one of the areas that we're most interested in is looking for biomarkers, for ways to identify what puts people at risk for Parkinson's disease and how the disease itself progresses. Todd, I know that you guys have an initiative underway to bring together industry, to bring together academics, to look for these sort of biomarkers. Maybe you could walk us a little bit through that program that you have and what you're getting from the information.
Yeah, I think just first to really discuss why biomarkers are so important for Parkinson's. We've heard the discussions at this conference and at other diseases like Alzheimer's, and how we need the biomarkers, particularly as we're trying to develop disease-modifying therapies so that we can really track whether we're having an impact on the underlying disease. In Parkinson's, we have sort of a unique challenge that results from one of our successes, because since we have a treatment, Sinemet, that can impact the symptoms of the disease, we can't just track the symptoms to look at the disease course and the disease progression. We need to really, because people are medicated, so we really need to try to get signals into the underlying disease process to know whether a drug is really impacting that progression. What the foundation has done is we launched an initiative that's called the Parkinson's Progression Markers Initiative, which is a large-scale study to look at the natural history of Parkinson's, starting with newly diagnosed patients and tracking them over the first 3 to 5 years of the disease, both clinically, but also with neuroimaging and biosampling. And the goal is really to get a great understanding of the clinical progression of the disease. There's great variability in Parkinson's. Some people seem to be more rapid progressors versus slower progressors. The correlation between the motor symptoms with some of the non-motor symptoms that Michael mentioned. And then to see if we could actually correlate those clinical observations with both the neuroimaging outcomes as well as some of the biological sampling that includes spinal fluid and blood sampling. What's interesting about the study is that it brings together the foundation, academic researchers, as well as industry all to the table together to try to solve this problem, because we all know that if we have better biomarkers, it'll give us a better insight into how to develop disease-modifying therapies. So it's sort of a pre-competitive area where in advance will greatly accelerate drug development across the board.
Clemens, you do quite a bit of work in this area looking for biomarkers. Maybe you can walk us through what's emerging at this point. What do you think is the most promising, and how important it is to have some kind of an objective measure, especially for a disease like Parkinson's disease, where many of the symptoms can be subjective?
So just to sort of start from the Harvard perspective, we started an exciting new joint venture between the Alzheimer's neurologists and the Parkinson's neurologists here at MassGeneral and at the Brigham in 2007. And we decided we are going to join forces and build a longitudinal biomarker study and biobank, both for earliest stages of memory impairment as well as for Parkinson's disease. And that's the Harvard biomarker study. We have now enrolled 2,300 participants as well as controls. And we're following them longitudinally for 5 years with detailed clinical phenotyping, biospecimen collections. So it's a joint venture here, but the cool thing about it is it's also an open resource. So it's open for collaborations, and it's an open resource also because we ask everybody who works with the Harvard biomarker study to search for new markers, to then input their data into our data set and our database, so that we can build on each other's research going forward. And in this fits, this actually starts putting together the pieces of a pipeline for discovering biomarkers, for validating biomarkers in the PPMI study, so that now you really, if you have a exciting idea for a new biomarker, instead of conducting a discovery study and a replication study, that will take you 10 years to do, enroll more than 2,000 folks, cost millions of dollars. You can do this now in several weeks. You can do the discovery in the time it takes us to take the samples out of the freezer and you to run the assays, and then you can knock on Todd's door to replicate it in the PPMI study. And that's an exciting thing, that there's a pipeline now in place to really very rapidly discover and replicate any kind of biofluid biomarker for Parkinson's disease in a matter of weeks, as opposed to years as it needed before, and just at basically assay costs at this point, as opposed to $1 million costs. Where's the field on biomarkers?
You know what? I'm sorry, Clemens. Can I just interrupt and ask both of you guys? Yeah. Are patients who are involved in this, are they asked to not get treatment right away, or do you track whether they are, when you're looking for their biomarkers and progression over time? How do you control for drug use?
Yeah. So at least and Michael could comment on this too, in the PPMI study, as people started early in diagnosis, many Parkinson's patients are not treated when their symptoms are extremely mild. So in the study, we actually allow the natural treatment that the patient and the doctor will decide on. What's unique about these studies is you actually do wind up getting sampling before and after treatment. I think the results from the PPMI showed that about 1 year in, about 50% of the patients still were not treated. So, that is interesting also because that might be a surrogate type of marker about the symptoms, you know, how much the symptoms are impacting the person, whether they felt the need to get treatment. But it does vary in Parkinson's, but it's not necessarily the kind of disease where at diagnosis people automatically start treatment. Now later in disease, once people pass that 1 to 2 year part, most people are treated, and you'll have to build that into the size of the study, make sure you're recording the information about the treatment. We're hoping to have biomarkers that are kind of independent of these dopamine-based treatments so that we really can track the underlying disease.
Clemens?
Right. So, and you wanted to also get a glimpse of where the field is, right, in biomarker discovery. So if you had asked me the question a few years earlier, the answer would have been simple. It would, zero, nothing, a blank slide. But actually now, the times have really changed, and the field is really breaking through in an incredible manner. The DAT scan was FDA approved a year ago, so it's a FDA approved differential diagnostic aid for Parkinson's disease. And we have biofluid markers, at least 2 of them or 3 of them that have been replicated in several independent large studies. Uric acid, for example, Michael has done a lot of work on that. Vitamin D and alpha-synuclein in spinal fluid have been found over and over again to be abnormally expressed in Parkinson's patients. So we have strong signals. The strongest signals in terms of biomarkers and really in a way revolutionizing the field is from the genetics. Now we know for sure that there are at least 24 risk loci for common garden variety Parkinson's disease. And so those are genetic risk markers for the disease that allow us to build up on hard data points. So these are, do we have the ideal biomarker? Certainly not. In fact, I think what has held the field back for a long time is that the question was phrased wrongly, that everybody was looking for the ideal biomarker. I think there is never going to be one ideal biomarker. I think that's completely unrealistic. In reality, we're going to have maybe 20, maybe 30 different biomarkers for different specific targeted questions. For example, if we study alpha-synuclein-directed medicines, we will have synuclein markers in the blood and in the spinal fluid, and hopefully also imaging markers of synuclein to measure response and pharmacodynamic effect for these treatments. And we will have markers for different subtypes of patients. And I don't want to take up too much airtime, but we'll come back to this later.
And I just to highlight on the broader picture, as we're excited to get to specific biomarkers, and a few are coming to the fore, I just wanted to note that you asked at what stage are people so-called de novo just starting, and as Todd was pointing out, in the PPMI, that's how patients start. It's a 5-year study, so that they often don't end up that way. Whereas the Harvard biomarker study is a little bit further in the disease. And these are just examples of tools, and I think very complementary, that the way the field is identifying that to advance, to do better clinical trials, to identify better compounds, that we need to build an infrastructure, and these biomarker cohorts are really infrastructures to get better leads and better ways to test the leads. So it's part of a sort of a broader community approach, and a great example how seemingly 2 different biomarker programs are really actually quite complementary and helpful.
We are open to taking questions. If anyone has questions, please send them in. We have the Yorn app. You guys can send it in on your phone. We'll take any questions. So Frank, maybe you can tell us how important these biomarkers are for you, having some insight in terms of what you guys are going after, and maybe you can tell us about ISIS Pharmaceuticals' Parkinson's program.
Sure. So we as a company, are really leveraging the genetic information that Michael just described, in that there are a number of forms of Parkinson's that clearly have a genetic component, alpha-synuclein and LRRK2 being 2 examples. where they're dominant genes, and it's thought that if you could reduce their function or reduce their expression, it would have a beneficial effect. And so we have a technology that we've developed that directly takes that genetic information, and we can use it to design our drugs, which are called antisense oligonucleotides. And what they do is bind to the RNA that encode these proteins and prevent the proteins from ever being made. And so it's a technology that we've developed that has a number of other drugs in clinical development, and 2 drugs that are approved for commercial sales based upon the same technology. So we have a lot of clinical experience with the technology, including within the CNS and delivering these drugs to brain structures. What's been challenging for us is because we are administering these drugs as an intrathecal injection, so it's basically a lumbar puncture, is being able to detect the pharmacodynamic effect of the drug. So are we getting enough drug to the right cell types in the body to be able to assure us that we are producing the desired pharmacology? And I think that's where some of these biomarkers are really going to be critical, is not only for disease progression and being able to monitor are we having an effect on the disease progression, but even more proximal, are we having an effect on the target? And to me, alpha-synuclein is, I think, a clear path forward that I think it's fairly well validated that we can measure alpha-synuclein in CSF, and we can measure it in systemic circulation. And that with our technology, we should be able to modulate the expression of alpha-synuclein either in these biofluids. Some of the other targets that we're interested in, I think, are a little bit more challenging, and I know that Michael J. Fox is investing a lot of time and effort trying to develop additional biomarkers for some of these other therapeutic targets for the disease.
So that, I think, is a great question, raises a great question, which is, do we know exactly what we're going after in Parkinson's disease? Are we going specifically for alpha-synuclein? Are there other targets that...
I think there's a number of different targets. So, one of the attractive features for LRRK2 is that there's a genetic population where I think we can do a very prescribed clinical study within that genetic population, seeing if we're having an impact on the disease, and then broaden it out to a sporadic or a general population of Parkinson's disease. And with alpha-synuclein, again, it's a target where I think there's an overwhelming body of data that suggests that alpha-synuclein is contributing to the pathology in Parkinson's disease. So it gives us a very, as well validated as you can at this stage without having therapies out there, a validated target to go after. Mm-hmm. But it's not to say that these are the only targets. I think there's a number of additional very, very good targets that one could think about for treatments of Parkinson's disease.
Do we have an issue in Parkinson's disease like we do in Alzheimer's with the amyloid hypothesis? Is there any disagreement? Is there any uncertainty that once we finally do have something that should be disease modifying, that perhaps is hitting the target, that we'll finally get the answer of whether it works or not? Or do we know for sure that it's the target?
I guess I'm in the center.
Yeah, you're in the center.
So as I said before, this is not Huntington's disease. Even in Huntington's disease, where there is one target, there are still many targets, and there's uncertainty. So yes, there are many genes which are some of the best clues that are being targeted. It's a landscape, and I think what's different now than the way trials were done 20 years ago for swipes of disease modification is they're done as rationally as possible, both with target identification and on the clinical trial science, but even on the target identification with LRRK2, as Frank was saying, being a leading candidate. And that theme of seeing a path where a rational approach can be laid out, there's infrastructure being laid, again, particularly by the Fox Foundation, in looking forward and sort of setting the table for that trial once the target is understood well enough, and actually LRRK2 patients, families, cohorts is actually being assembled to learn, within the biomarker framework, but also to be ready in this anticipated convergence. So there's a lot of angst still, of course, because we don't have a track record to rely on, but it's also very clear that at both ends of the pipeline, identifying targets and how we prepare and conduct the clinical trials, that I think we're going about it a better way, but we still have to get there.
I think also there is no unified hypothesis of Parkinson's. So there's a lot of clues. There's LRRK2, there's alpha-synuclein, there's some of these other genes, Parkin, PINK1. there's inflammation. And I think what the genetics is giving us more direction to is, can we actually start to piece these together and put the puzzle together? And it might be that there's different puzzles given the variability in Parkinson's. But I think that's some of the basic work that still needs to be done, and it's gotten a big push forward because of some of these specific genetic changes that we know can lead to the disease and how they now intersect with some of the other pathways that might be involved in the disease.
And also, in that uncertainty, as we drill down where the clues are, and working with each of the lead genes, for example, there's a path to targeting anything that's specific about that, but there's still not just a hunch and a hope, but there's clear evidence that these clues are also converging on theories that have been around for a while, but leading to more tractable targets within them, as mentioned, inflammatory mechanisms, oxidative damage, proteasomal toxicities, and the like. So we're still at a very early stage, but between the refining of the clues and the structures that are being developed, I think it's a much more hopeful time.
So we have some clues, some genetic clues like the LRRK2 and GBA, I believe, a couple of other things that appear to be perhaps causative in Parkinson's disease. Then there are also other genes that seem to just increase someone's risk for Parkinson's disease. Where are we in terms of translating those kind of findings?
Right, so that's really all what we are about, to take the power of genomics and apply it to Parkinson's care and use it to change the way we diagnose and treat and perform clinical trials. And so one of the big goals really in our laboratory, and what we're doing now, is to systematically dissect the genetic architecture of prognosis in Parkinson's disease, to identify all genetic variants that predict fast progression or slow progression, that predict who will develop dementia in 5 years and who won't, who will be in a wheelchair in 7 or 8 years and who won't. And if you have these predictors, what we're seeing, what is emerging really from the data is that you will be able to see genetically defined subtypes of Parkinson's disease that have a very different disease progression, and that you can then use this information to enroll a genetically defined homogeneous trial population that's probably smaller than if you would use the all-comers, and it has a more homogeneous progression, thereby again saving you dollars and sample size.
Did you have something?
No, I think there's a lot of lessons in neurodegeneration, as was mentioned earlier, in the oncology area, where again they're identifying therapies that are really cell signaling cascades that are being impacted within the therapy, and they're identifying genetic signatures that inhibiting a RAS pathway or inhibiting a JNK pathway or some of these other signaling molecules might have an impact in a subpopulation within the therapy. And I suspect it's going to be very similar in these neurodegenerative diseases too, where there are genetic signatures that may point to novel therapies that aren't necessarily genetic causes, but they're impacting the pathways that are related to the genetics.
So we have a question here. Do you anticipate one assay to catch all biomarkers, and how would that tie into patient costs? So I guess with some of these, some of the work that you guys are doing now, do you envision one test, one assay to find all these biomarkers? What are you looking at now?
So I envision 50 biomarkers or 100 biomarkers, and will be it's hard to see that one assay can handle this. And in fact, some of these markers are likely to be protein-based, CSF, alpha-synuclein protein. Others are metabolites. Others are gene tests. So it's not going to be one assay. It's going to be several assays. And how is this going to work into costs? Thankfully, that's why I didn't go to Wall Street. We're not here to do quarterly results. We're here for the moonshot. for doing breakthrough science. So we'll worry about that later. Yeah.
So Clemens, do you have to get cerebral spinal fluid? Can you do it with a blood test? Are you looking at urine? What kind of?
So I think you want to keep an open mind and look at all of the above. CSF makes a lot of sense because it's the brain floats in cerebrospinal fluid, and the neurons in the brain exchange the metabolites and proteins and RNAs with the spinal fluid and microvesicles. So it gives you a relatively easy access to the brain environment. So it's
I don't know about easy.
Well, so we are encouraging all our Harvard biomarker study participants to have a lumbar puncture, and so what we try to remind them is of maybe you had a child a while ago and got an epidural, right? So that was a happy experience. The epidural actually did good for you, and a lumbar puncture is not much different from an epidural. So it's a routine procedure, and complication is very rare, headache in about 1%.
And PPMI is sort of reset the standard for reintroducing that needle comfortably. Is it 5 or 7 in a row that?
Yeah, people in that study will get up to 5 or 7 lumbar punctures, and there's been a pretty significant retention of people in the study. One other just comment to this question, I think, is what we know from Alzheimer's disease is that there are different biomarkers also that are useful at different stages of the disease. So there might be something that would be useful to help with a diagnosis or a genetic test that may determine if someone's at risk, maybe very different than something that's useful later in the disease to determine whether they're going to go on to get dementia or some. Yeah. So I think that it's very unlikely there'll be one test across the whole spectrum of the disease and also within the whole variability of the disease.
And in casting a wide net, it's also the idea that more peripherally there may be clues that's supported by this broadening evidence that the disease may in fact be a more systemic disease, and certainly the blood work that these teams are doing are supporting that. And then you'd like to be where the cells are degenerating, and biopsies aren't a real option in this disease, happily. But neuroimaging is advancing and is our direct window into that, even though it's not a wet marker, and it remains one of the more exciting but still unfulfilled biomarker realms.
We're also finding alpha-synuclein is expressed in saliva. So
and
I think
skin.
We have so very broad in colons, so even in some colon biopsies you can find alpha-synuclein. So there is a very kind of open mind approach to where we might find the biomarkers.
The exciting thing is that as you hunt for biomarkers, you actually learn about the disease process and the disease biology. So for example, we more than a decade ago found extremely high levels of what until then was called the neuron-specific gene alpha-synuclein in blood. And it turns, digging into this very mind-boggling finding over several years, we and others found that synuclein is actually a erythroid specific or predominant gene, predominantly expressed in the red blood cell lineage. That's also expressed in neurons, but much lower levels. But it's relatively specific to just those 2 cell types. And certainly that's going to tell us something about the normal function of alpha-synuclein. But it also gives us an avenue to monitor it as a biomarker in the red cells, perhaps parallel some of the processes in neurons. And a similar story, is developing with LRRK2, for example.
Before you go there, can I just, I'm sorry to interrupt you, we'll come back to LRRK2, but that makes me a little nervous if you say that alpha-synuclein is in the red blood cells. And if we're targeting alpha-synuclein, going after it, are you sure we're not going to have some unintended toxicities there?
So we're targeting in the CNS. We're directly delivering the drug into the CNS. Some of the other approaches for targeting alpha-synuclein would lower it systemically as well as centrally, but it remains to be seen within the red blood cell whether the antibodies are getting in.
So right now there are actually some studies that have gotten into the clinic for immunotherapy against alpha-synuclein. And I think we're talking about alpha-synuclein as the target, but actually there are many forms of alpha-synuclein. So alpha-synuclein forms aggregates in the brain, and it also exists as a monomer. And we believe, and some people say they've detected, forms along the way, fibrillars, oligomers, to the aggregate. And I think at least the current goal of the immunotherapies is actually to try to target more towards the aggregated or pathological form of alpha-synuclein versus the more native form that might be found in blood. But this obviously is something that has to be tracked based on the therapeutic approach as a potential safety monitoring. It's there for a reason, but hopefully we can develop more targeted therapies against really the path. Even in the brain, it's there for a reason, so you don't want to just, but it is overexpressed, we believe, in Parkinson's, so it might just be bringing it down to a normal level. But this is something, that's what has to be done in the clinical development plan.
And you were asking about controversies like Alzheimer's with amyloid versus tau. While I think in the Parkinson's field it's actually very harmonious, that's probably a good area where there are people who heatedly debate whether you should be targeting the clump protein or these days more what stage of the earlier protein. And back on the safety concerns of something that might be expressed systemically, I'll just bring it up because I've seen the work of I think you've been involved, Todd, in the Foundation for the LRRK2 target, something that gives perhaps extra rationale for the direct targeting in the CNS that Frank's talking about with the antisense technologies, because in being very careful after getting excited about something that's clearly relevant to the pathology and trying to find a drug, but having to get past the parts outside the brain, it's important to be very rigorous and systematic, and that has turned up some source of pause for LRRK2 in the pulmonary and renal systems, and I don't know where things are at now, Todd, but that's...
Yeah, we've done more work on that. We actually think it's surmountable and that it's not a significant issue, but LRRK2 is also functions in these other regions, and what's it doing there is an important question. But you have to, test it against each drug and be very rigorous. But we had a very interesting follow-up on this where we had a collaboration between multiple pharmaceutical companies participating with their compounds to really try to dissect this potential safety liability. And I think it's a great example of the companies sort of stepping forward and releasing that data more broadly to solve a general problem in Parkinson's. And it's allowed that field, I think, to kind of be reinvigorated based on where we were a year ago.
So let's stick with that for a minute, and Clemens, I know that we're going to come back because you had a point to make about LRRK2 as well, but Todd, maybe you can walk us through that, the fact that the pharmaceutical companies did come pretty openly to the foundation and shared their data. Is that something that you're seeing more of? Is the industry more open and collaborative at this point, and how can that continue going forward, especially once we start getting to actual therapies?
Yeah, I mean, we've seen that in Parkinson's disease, which I guess is where I can comment the most, that there are still a lot of really fundamental questions around biomarkers, around how these genetic targets are really contributing to the disease process, that we've seen great examples of collaboration in the industry around, because that information will help everybody. I'm always a big fan of these kind of collaborative efforts when companies share their data, work very openly with academics. I'm also a big fan as a proponent for new therapies for Parkinson's, for the day when the parts that they don't want to tell us and they're competitive on also, because that's sort of the motivation of moving forward, and that means they're onto something. But I think there's been a great balance sort of in Parkinson's around these pre-competitive areas, open sharing of data, and I think the foundation kind of plays a good role in being a neutral venue for that, and bringing academics to the table to solve these problems as well.
Clemens, I'm sorry I interrupted you earlier about your comment on LRRK2. Was there?
Oh, LRRK2, I just wanted to sort of point out this interesting story there. So they have a LRRK2, a Parkinson's gene. It's both a familial Parkinson's gene, but it's also a risk variant for common sporadic Parkinson's disease. And turns out that it's actually is expressed in neurons to some degree, but it's very highly expressed in white blood cells, specific white blood cells, monocytes and lymphocytes. And so it really invigorated the area of neuroinflammation research in Parkinson's disease. There are many clues from epidemiology that had pointed at a role for inflammation in Parkinson's disease. If you take a lot of ibuprofen, your risk for Parkinson's appears to be lower, for example, and we knew from looking at the brains that there was evidence of reactive microglia activation in the brains. But you know, those were sort of more softer correlations. But now with the genetics, there was a clear, you know, clear data pointing at LRRK2 and its expression in white blood cells, its role in neuroinflammation. So I think this area is really now on, you know, it's really taking off, and I think great things will come from it.
Great. And Todd, have we put to rest the concerns that we saw in those early animal mouse studies?
I think that, you know, to rest, they still have to be, you know, evaluated and will have to be tested, and no LRRK2 drug's gone into a person yet. So we just need to make sure we're aware of what we're looking for, but I think that we have a better sense for what to look for and maybe how to kind of manage that going forward.
And there are things that you can monitor to help assess what the potential for toxicity, and maybe pick things up very early in the process.
So we're going to switch it up just a minute here. Michael, you were talking earlier about how we're having a better understanding now of Parkinson's disease and how it seems to be perhaps more systemic. It's not just exclusively a motor neuron disease. We have a question about the non-motor symptoms of Parkinson's disease and how they progress and if it affects treatment at all. So wondering if you could take that.
Sure. There's the clinical end of that, which is the reality for Parkinson's patients. And then there's the insights that leads to, actually, which we haven't gotten to, over prodromal, premotor Parkinson's that's opening up new opportunities. So I'll just mention, as we were highlighting before, that Parkinson's is much more than these motor deficits, and the disability of Parkinson's is often overshadowed in the form of whether it's depression, dementia, psychosis, sleep disorders, even some of the systemic issues. And in some cases, you know, we do have symptomatic therapies, and it's good to identify. Some patients will just focus on that tremor when they can't get up because of depression. So it's good not to ignore that with your family and your physicians and the like. So we can talk more about what that breadth is, but I just also wanted to bring it back, since we hadn't touched on it, this concept of prodromal that's also extended out of the synucleinopathy clues, for example, and how it's even changing not only how we view the natural history and the pathophysiological progression of the disease, but also shifting a little bit how we think of, for disease modification, ultimately how we might develop clinical studies. For years, and if this is still the case, sort of the gold standard for disease modification trials is looking for the earliest Parkinson's disease so you can nip it with your best shots as early as possible. But with the appreciation that disease features even precede that, now there's efforts to characterize better in the years, decades even before the disease starts, and its motor deficits, how that manifests. So it was mentioned that there's smell difficulty with smell, constipation, or also a characteristic sleep disorder called REM behavior disorder. And it's been appreciated that if we can get a better understanding of what defines the prodromal phase, we may be able to start our interventions earlier. And this is just an example of many of the other pages that we've taken from other neurodegenerative disease initiatives and the like. So MCI, while that's still cognitive, precedes dementia as an example of something that is now the focus of many disease modification efforts in Alzheimer's. And we hope to get there with Parkinson's. Ultimately, of course, there's prevention in the context of even a defined genetic clue like LRRK2, someone who has the gene, just like someone who has a Huntington's gene and knows they are a risk. We'd like to prevent it, but we want to concentrate where we can best define things with our best leads. And that non-motor feature has opened up this new concept to looking to how we can try to stop the disease.
One of the other things that I think the better appreciation and assessment of the non-motor features of Parkinson's has done is changed our whole thinking and concept about the progression of the disease. So the progression of the disease has always historically been defined by the motor symptoms. When we say someone is early Parkinson's, that's really defined by the motor symptoms. That's what probably brought them to the diagnosis. But we know from studies like PPMI and other studies that someone could be early motor, but actually quite advanced in their non-motor symptoms, and vice versa. There could be people who are fairly late with their motor symptoms, but not be as affected by the non-motor. But it's a holistic disease, so I think kind of getting a better sense for what we really mean by the progression and stage of the disease. The traditional staging process is really based on the motor symptoms, the 1, 2, 3, 4 staging. So I think there's been a lot of work in the field to better integrate the non-motor features into that sort of idea of the staging of the disease. Much more work to be done though.
So when we talk about, the topic of our panel here is about the therapeutic breakthroughs, the advances that we're having in Parkinson's disease, and it does seem like when you look online, the pharma recently put out a report that said that there's 37 drugs in development for Parkinson's disease, 43 clinical trials out there, and they are kind of across the board. Some of them are genetic, some of them are symptomatic relief. When you look for your patients, I guess my first question would be, how do you advise people, how would you advise people to find out their risk for Parkinson's disease? And if they have early stage, if they can be tested for LRRK2, is that something that, given we don't have any kind of disease modifying therapy at this point, do we, to what extent are you pushing to find those patients?
Right, so that's tough question. So it's tough from the clinical side, it's easy from the research side. So really, time is really now to do the research on genetics and how we can use them. So we would encourage them to enroll in Harvard biomarker study, PPMI, and then we will check their genome for variants and use this information to find new biomarkers, new targets. Now, if this patient just comes for a routine visit, so the question does arise, and well, let's make this real. So, the young patient comes to the clinic and has children, and says, I want to know everything about my disease, and I want to do any kind of clinical trial that there is out there. Participate in fundraising, anything, and so then we wanted to have the known familial Parkinson's genes checked in this patient, which actually costs $15,000 if you pay the bill out of pocket, which is a lot of money. Now, what happened? His insurance said, well, we're not going to pay for it. But fortunately, he had a patient advocate and a genetic counselor who were really on his case and managed to convince the insurance that because it's important for his life planning and decisions in view that he has children to know whether he has one of the familial genes, the insurance then agreed to check the autosomal dominant and recessive Parkinson's genes and then cover it. And so we ended testing for that.
If patients...
Just going to add that I do think this is sort of a evolving environment around genetic testing and what people want to know and don't want to know. There's been a very significant Parkinson's study that's ongoing with 23andMe, given their interest in Parkinson's. And for many years, Parkinson's patients could participate for free and get all the results. And it was up, no one forced you, so some people decided to do it and some people didn't. So I think that there are some people that want, like the example Clemens just gave, that really want this information. they want to act on it and, you know, they want to know everything, and there are some people that aren't interested. But right now, there's nothing that would change about your treatment by knowing, but it is, you know, important from a research perspective, and some people, they want to really know, and then they might change some things they're doing in their life, try to exercise and eat better and do things now instead of later.
They can do that.
Yeah.
But I think we are really at the pivotal moment in our society where things are going to change. At the moment, we still have the division between, you know, doing the research genetics and not telling and you're not allowed to tell the patient anything. I think this is going to change. Patients are, medicine is going to change. It's going to become more participatory, and patients will take ownership of their genes just as they take ownership of their scans and their other information. And so I think that wall will certainly crumble to some extent over the next years.
But just to return to your initial statement, Clemens, that it's clear on the research and I think it's also for therapeutics or for managing one's own Parkinson's when people are struggling with a new diagnosis. I don't think it can be emphasized enough how valuable it is to if you have siblings and you're Ashkenazi Jewish, you may be a great candidate for the LRRK2 cohort, for example. That would be an example where finding out the genetics, or at least participating along those lines, could be very valuable. But even for the non-genetic opportunities, participating, whether it's a blood test study like the one Clemens overseeing, observational study, it's very empowering for patients to try to take hold of the disease. And one of the things they can do, whether it's helping them or their children or just other people, is to get us to who knows where we'll be in 10 years, but only if people participate. So I think it's sort of a side answer, but one of the strongest rationales for finding out more.
And that's interesting because in much of the stuff that I have read discusses how important it is to try to get patients enrolled in clinical trials. Is that at all a struggle for you guys to get patients enrolled? And how do people pick? Because fortunately we are in a situation right now where there are a number of trials underway. So I'm wondering, you know, is it a problem if you start drug treatment? Does that rule you out of clinical trials? How do you advise your patients once they're diagnosed whether to, you know, do a tried and true, you know you're going to get some benefit if you go on levodopa, or do you roll the dice with a clinical trial, and how do people decide?
Well, I'll just speak to one of the ways in which the answer is unfortunately yes. There are parts of Parkinson's progress that are clearly limited, bottlenecked by enrolling patients in Parkinson's trials. Partly it's because of this impetus to try to, when we have only a few shots at this point, you mentioned there were 35 or 40 some trials. Only a handful of those at this point are disease-modifying trials. We haven't really talked about the low-risk therapeutic improvements, which are great, but for disease modification, okay. For disease modification, you know, we're on soft ground. We're trying to do the best we can with the best targets and the best clinical trial designs. And for now, the best clinical trial designs are as early as possible. These so-called relatively rare de novo subjects. And they're...
De novo meaning untreated?
Yes, de novo meaning untreated or largely untreated, so just recently diagnosed and without the complexity that Todd mentioned about the confounds that the very effective symptomatic therapies introduce. So yes, trials have taken years, tens of millions of dollars longer to get to their answers so far. And it's sort of, again, in the sort of community-wide and infrastructure level, it's been seen as a challenge. And some of it's just mechanical, because most generally people, for the reasons I mentioned, want to contribute, but they might not know about trials, especially if they've just been diagnosed and they're reeling and they're trying to figure out what good resources. It might take a year before they're referred to a neurologist or a movement disorders neurologist, and the window for their candidacy as a de novo patient might be lost. So again, on the lines of infrastructure, NIH and more recently, particularly the Parkinson space, the Fox Foundation with foxtrialfinder.org is short-circuiting that and helping. It's still a challenge, but the community is mobilized over that.
I think, you know, just to add to that is that, you know, we study a lot of different rare diseases, and we're surprised how how hard it is to inform patients of clinical trial opportunities, not just our trials, but in general. And a lot of times the primary physician who's taking care of them is unaware of all these activities and tries to take care of them. And that's where organizations like Michael J. Fox do a wonderful job of advertising what's out there for the patients, so that they really have to do the research on their own and figure out what's the path that they want to follow, and then go to their physician and ask them about it.
So one area where patients definitely want advances is in symptomatic relief. So let's talk about what medicines are in development. We've had some recent success in terms of, longer acting drugs, different delivery mechanisms for more severe patients and whatnot. So for patients who already have Parkinson's disease, who maybe aren't getting enough benefit, what do we have now, and what do you guys see as, in terms of the future, what's promising coming up?
I think one area where there's been a lot of recent advances is around the delivery of levodopa. So we've talked about levodopa being the gold standard treatment for a number of years. As an oral medication that has very unique sort of absorption and metabolism, it does have some limitations. And as the disease progresses, people with Parkinson's who are taking levodopa will have what we call motor fluctuations throughout the day, periods where the drug works and comes down and they're on and off, and those can become much sharper, where people are taking very frequent medicine every hour or two during the day and balancing periods where they're on to almost frozen. And there's a hypothesis that's been, I think, supported somewhat with clinical data, that if you can even out that exposure during the day of the drug so that people don't have the peaks and valleys that come from an oral medication, you can get more on time with less off time, and without these side effects that are dyskinesias, where you overshoot and actually have too much movement. So there's some therapies looking to develop improved delivery that would get you more of a continuous exposure. We're also seeing some improvements in more rapid onset. So if someone with Parkinson's is stuck in one of these off-frozen periods, right now they basically take their medicine, have to wait for that to kick in, which could take 30 to 40 minutes. And there's some versions of levodopa being developed that are inhaled versions or sublingual versions of dopamine therapy that could have much more rapid onset, so that while you're waiting for your drug to kick in, you can get that benefit. So there's been a lot of, this is really leveraged work outside of Parkinson's that have focused on drug delivery technologies, and being that we know levodopa is an effective drug, it's been a good candidate to apply to these technologies.
I'll just mention too, just sort of the broader view of the drug development for Parkinson's. We started sort of with the holy grail of trying to stop or slow the disease or to reverse it. We haven't gotten to regenerative kind of therapies on the one end, which is really high risk, but what everybody wants to see, and what we're clearly getting closer to on the one hand. And on the other is the 50-year-old therapy that I have to say, in the last 10 years, I've been impressed, having viewed that as sort of rearranging the deck chairs between levodopa and dopaminergic agonists. But it's very striking, actually, some of these recent advances in delivery, how much it can just help the quality of life because of those fluctuations. There's also a middle ground that hasn't quite delivered as much, but it's still active, which is symptomatic therapy that try to take a different approach, non-dopaminergic therapies. There's various other transmitter systems besides dopamine that's misregulated that can affect the motor symptoms, cognition, and the like, and whether it's glutamate or adenosine. There are actually a number of clinical trials, even at phase 3 now, that are ongoing that haven't delivered as much as we thought, but they're still promising enough. So there's sort of this spectrum of a very effective, worthwhile, symptomatic, even dopaminergic therapies that are still yielding advances across the spectrum towards this more hopeful but uncertain area.
It seems to me like Parkinson's disease is lucky in the extent that there are some very high-profile patients, Michael J. Fox, Sergey Brin, who are willing to put their names and their money behind some of the research. So we have these foundations. And it seems like the industry is now, as we're getting more information, genetic information, coming on board as well. Maybe you guys can talk about that in terms of, it's been 200 years. 100 years longer, twice as long as with Alzheimer's disease. We look at these advances that we've had in cancer and heart disease. It's really remarkable in the last 15, 20 years. Is it time for Parkinson's disease, do you think, with the foundations, with the industry? Are we at an inflection point? Todd?
Yeah, I mean, I think we are. I think we have really, on that, if you graph the knowledge of understanding of the causes and underlying mechanisms of the disease, we were kind of like this, and now the last 10 years is kind of really swooped up exponentially, and there is more resources coming to the disease. It's still minimal compared to cancer when you talk about cancer. So I think there's a lot more that could be supported in this area, but we are seeing excitement around some of these targets. Big pharma is getting involved and engaged. There's a robust, academic inquiry in Parkinson's. I would like to see more funding coming from the government, in this area across the board, particularly around trying to put the pieces together around the mechanisms of the disease. But I really do think we are now at this, we're seeing those first genetic-based therapies getting to the clinic. It's really it's sort of a new world for potential Parkinson's therapies than we probably didn't envision even 10 years ago.
What is the timeline? When might we start seeing new therapies, completely novel therapies, as opposed to tweaking what we have already? And to what extent? What are we hopeful for? Obviously everybody wants a cure or better prevention entirely, but what are you really aiming for at this point? At what point would you consider it a success? How much do you need to slow the disease by?
Well, I think that it's realistically what we're really hoping for, and I think with some realism, is to slow down the disease. A cure is certainly possible, and we're working towards that, but I think the several-year horizon is to slow down the disease. I think there's a tier, as there has been actually for a decade, of sort of the best candidates in a handful of trials aiming for disease modification. Nothing has worked so far. The trials are being designed better. I think the bets are better, and there are several that are ongoing now. It's quite possible that any one of them, NIH has invested in a calcium channel blocker based on some strong biology, isradipine, that's now being widely tested in North America. And there are other trials that are coming online like that. I think any one of those could yield the fruits that we're waiting for. I think this sort of next wave of the genetic explosion leading to clues is probably on a 5 or 10 year timeline, because unfortunately we didn't get into fully the bottlenecks of the clinical science, but once you get that great idea, unless it's going to stop the disease, and I think realistically we're looking for, generally these are powered to slow down the rate of progression by some measure or another between 20 and 40 or 50%, which would be a tremendous change. But that means you can't just give the drug, look 2 weeks later, and you have to test it even in a large number of subjects over years, and you have to have enrollment for that. So you talk about 4-year projects to do a well-designed, typical disease modification trial. So that's why I say there's some things in the pipeline, and there's some even more exciting things. And just to get back to your original question about this, there's no question that it's exciting, and that to get there, we have to invest in it, and it's hopeful that there's more coming. It's still a drop in the bucket compared to other approaches, and I'm confident we'll get there. How fast we get there is really what will be determined by the investment, by society, by people who step up.
So we've only got a few minutes left here, and there's so many things to discuss in Parkinson's disease. Let me just go through a couple of these questions quickly. Do we have a current theory about the role of vitamin D in Parkinson's disease? Is that a hot topic? I have to.
That's a very interesting story that sort of smoldering for a while. Well, for sure, everybody knows vitamin D helps your bones, and that's good for Parkinson's, right? They are at high risk for falls. If you can take vitamin D, you can cut your risk of bone fractures in half. But that's useful for patients, but what's really exciting about it is that dopamine neurons are full of vitamin D receptors, and there are interesting research data to suggest that vitamin D actually might be a kind of growth factor for dopamine neurons. and it's protective in several animal models of the disease against dopamine toxicity. So it's an interesting story.
All right, I think we've only got a couple more minutes here, so maybe we'll just wrap it up. I don't know if perhaps we can start with Todd and just go down the panel in terms of what your hopes are for the next couple of years with Parkinson's disease and maybe what you're looking for next from industry, from academia, from researchers here in the room.
I guess I'll go just since it hasn't really been mentioned, and I do think it's really really accelerating our progress, and it follows a point that Clemens was making, was kind of this intersection between research and care that's starting to be broken down. And what we're seeing from the foundation perspective is sort of a new invigorated role that the patients are playing in the research process. I mean, we have in Fox Trial Finder, for example, we have almost 50,000 people that have signed up saying, I'm willing to participate. I want to be part of this solution. And I think that's also something that's kind of changed in the last 5 years and will give us better insight in what it's like to live with Parkinson's and the effectiveness of the medications. So I think that's something that I'm very hopeful about going forward, and how the intersection of the patient component with the research component will really lead to greater breakthroughs.
Clemens?
Right. I think this is really a very exciting time for Parkinson's disease, and over the next years our view of Parkinson's is going to completely change. Instead of having this one-size-fits-all model of the disease that is largely based on 200 years of dopamine replacement, we are going to have genetically defined types of Parkinson's disease. And we're going to change the way we do clinical trials. We're just going to look at the specific carriers of a mutation, and we're going to do small proof of concept trials in these carriers using wearables, using biomarkers, and only then, when we see a signal in this new type of proof of concept trials, then we'll plug into the traditional phase 1, 2, 3 trials.
Michael?
As the clock ticked to zero, I was glad you mentioned wearables. That wasn't mentioned earlier as a new notion. But to step back to what Todd was saying, for me, what has me most excited is the sort of the convergence and the teamwork that's playing out. The patients, both as a community being involved in the trials, as supporting development, the foundations, NIH, academic approaches, and sort of with a full court press on the team metaphor, really making things look hopeful for the next decade from my perspective.
Frank?
Yeah, I would just echo that this is a community, and it really takes a community to solve a problem like Parkinson's. And so I would also echo that, thanks to the patients, the advocacy groups, and the physicians that are taking care of the patients, and the basic researchers, that I think we have some clues that are going to make some strides in developing therapies that impact the disease course. I think it's the beginning of that process now. I think, with these clues, there are subpopulations of patients that I think we have a better chance of helping, and that's a path forward. And we'll learn from that, and it'll create more and more therapies as we're going along. So I think we're at this cusp where we're beginning to see some real progress going forward.
Excellent. Thank you all for your time. Thanks for your attention.